My screen is not visible yet. Yeah, now it's visible. Wait, let me- Okay, I hope I'm audible to all of you. Okay, I think we shall start. Already waited enough. So today's week is about enzyme production, in particularly cloning. And then we will see overexpression, and then we will look at purification processes later. But today we will talk about essentially enzyme production, how enzymes are produced. So for that, the idea is in enzyme production, we require large amount of protein. And this is for, let's say, commercial purposes, for anything, for industrial applications and um, uh, stuff like that. So the idea is enzyme, we already know what we need to purify. That is mostly a protein. So when we are talking about enzyme production, we mean proteins in general. We will find the corresponding RNA of that protein and the DNA, that is the gene. From that DNA, we will extract that DNA, put it in a suitable vector, and then we will go for overexpression. Then we will purify this protein, and we'll get the pure enzyme. This pure enzyme can be used for its structure determination, okay, where we can get the structure of, let's say, mutants of this enzyme or the wild type, or we can use it for, let's say, for catalysis for the enzyme to work, or we can go for some industrial application. So this is the basic flow chart that we have for enzyme production, and this is how it is usually done. Now, if I talk about the schematics for producing enzyme, we need to first identify the gene that produces that enzyme, right? And let's call it as gene X, right? For that, we need to perform screening. And how is screening performed? We will talk about it in details. After the identification is done, we will clone this gene in a suitable vector. So for that, we require gene amplification. And the most preferred method of gene amplification is PCR. We'll talk about it as well. Then we will clone it into a vector. I hope you are familiar with these words, these terminologies. And then we will screen the clones for finding the clone that has the enzyme that we want. Following which, once we are done with cloning, we will go for overexpression. So for overexpression, we have to find a suitable host where we will actually, you know, um, purify this or overexpress this enzyme. Host could be very frequently used. Host is E. coli. There are different strains of E. coli for different purposes, very much used. Apart from E. coli, yeast is also used. It's the lowest eukaryotic organism. We also use insect cell lines and mammalian cell lines as well. The problem with insect cell lines and especially mammalian cell lines are that they it's difficult. It's expensive. The media and everything else is expensive. So overexpressing large amount of proteins is a problem. But why do we need those kind of cell lines? Because they are higher order cell lines. They are, I mean, in the evolutionary chain, they are a higher order organism. E. coli does not have all the modifications that are necessary for the enzyme or the protein to function. All post-translational modification systems are not functional in E. coli. In such cases, insect cell lines or mammalian cell lines are helpful. Apart from that, we go for transformation. Transformation is basically um, you insert your gene containing plasmid into the bacterial system. For bacteria, it's called transformation. For eukaryotes, it's usually known as transfection. Then we go for screening. So we want to see whether we have actually been able to successfully insert our gene, our plasmid into the bacteria. So for that, we need to perform screening. And the most used, I mean, screening method uses a selectable marker like antibiotic resistant genes. Now, these antibiotic resistance genes will essentially differentiate the positive colonies, and you can, further again screen them to see whether you have your gene. Then you can overexpress them and you can go for analysis where you will find whether your protein is present or not, whether they are expressed properly, whether they are being folded properly, whether they are in a form where they are soluble and functional. These kind of things are very important. Then we go for purification. Purification of this protein. Now you have, so in a bacteria, there would be several proteins that will be expressed, not only in bacteria, in any kind of host expression system, there will be many proteins that will be expressed apart from your protein of interest. So hence, purification is very essential. So for that, we will first go for cell disruption. So cell disruption can be done using a homogenizer or a sonicator or even bead beater in certain cases. So essentially, we will disrupt the cell and take out the proteins out of it. Then we will go for chromatography. So there are various chromatographic techniques which is based on different principles, which is implied for your protein purification. So, for example, if your protein has a histidine tag attached to it, you can go for a affinity chromatography and get the histidine. I mean, that could be one of the ways of purification. Then we go for estimation of the yield that we are getting of the protein, and then the purification analysis, how much protein you are getting, what is the purity of the protein, and all that kind of thing. So if we talk about the approaches for isolation of gene of interest, okay, so far so good. I would just require a thumbs up from your end. If everything till this part is clear, you can see the annotations. Yeah, good. Great. And listen to me properly. So again, for approaches, we'll look at all the approaches. And this has been discussed in detail in the slides as well. I'm just recapping all of this. So there was a pre-genomic era where not much was known about the, about genomes and genes in specific. So essentially, when gene sequence is not known, so what happened was we would take the genome and we will create a genomic library. So how is it done? Essentially, the entire genome is fragmented into smaller parts, and then we express each and every gene to see, or each and every fragment to see which particular region corresponds to our protein of interest. So the genomic library is nothing but a collection of clones. Otherwise, we can take the transcriptome. This is a little bit easier process because, you know, usually in case of genes, there are multiple regions which are not translated as well. But in transcriptome, we are only talking about mRNAs. So this is essentially the area, I mean, or the part of the genome which is always expressed. So you take the transcriptome and you perform cDNA. I mean, make cDNA out of it using an enzyme called reverse transcriptase. Uh, it had been discovered lately. That's why this approach was used later. And then this cDNA is used again to form the genomic library. While this approach is more preferred, but sometimes some regulators, some intergenic regions which might not be expressed as such as in form of protein could have a regulatory role in the protein production in a normal system. So those kinds of things can be missed when we perform transcriptome analysis. So anyway, in whatever situation, you get a genomic library, after which you perform screening. So what happens in screening? You will isolate your screen, which will have the clone containing fragment, and then you will get the positive clone, right? Now, in the post-genomic era, which is now, where genomics is very much accessible. Now we have, different techniques like next generation sequencing, uh, which which makes it very easy to sequence the entire genome of multiple organisms. So even you must have heard very recently that the genomes of several humans, around 10,000 humans have been submitted in a database, and that has been done from India. And there are several institutes which are involved behind this, several hospitals which are involved in this, and this is essentially to help create medicines and to understand the demography of Indian population in general. So this is the genomics era. And I think this has been also amplified due to the COVID-19 pandemic. Anyway, the point is gene sequence is known in this case. And once the gene sequence is known, what we do, we directly go for PCR. We can design PCR primers with this. So we go for PCR, and then we get the fragment which contains the gene of interest, and then we perform cloning. This clone again has to be screened, very similar to how we did earlier. So this is the essential steps. So coming to each and every step in particular, we'll discuss each and everything. So how is a genomic library constructed? Let's look at it. So essentially, we will first isolate the genomic DNA. Then we will perform restriction enzyme digestion. So that will basically, the entire genomic DNA will be cut into several fragments. Then we will purify these DNA fragments. And then what we will do, we will clone these fragments into, let's say, yeast artificial chromosome. Especially this yeast artificial chromosome is used for eukaryotic system. Let's say you are trying to get genes out of humans. So yeast artificial chromosome can incorporate large sizes. We will talk about all of this. So we will create a library of clones like this. Then what we will go for is transformation into yeast. So this is how it looks like. So this is the entire schematics if we look at it. So let's say you want to get human DNA and the vector is, let's say, yeast artificial chromosome, and you digest this human DNA, which will be a very large genome. You digest this with, let's say, EcoR1, which is a restriction enzyme, and you will get large chromosomes containing fragments like this. And then these are the fragments, and you have the vector, which usually looks like this. You will insert, you will also digest, okay, sorry, this vector with EcoR1. Um, so that you have eco-R1 site in this vector. Anyway, uh, just understand this is how it is. Then we will go for ligation, and we will get basically, a transformed plasmid, which are also called as the recombinant plasmid. We will insert this into yeast. So that would be the transformation process. And then we will get the genomic library. Okay, I think that is clear so far. Okay. So we talked about genomic library construction, then we will talk about cloning. How is cloning performed? So based on the size of the fragment that we want to insert, based on that, we select a suitable vector. Okay, we will talk about it. What are the different forms of vectors? So it is choose based on size. Um, so we already talked about it. The fragments are generated using restriction enzymes, and the vector is also treated with the same restriction enzyme, and then we go for ligation and we create the clone. This is how it is done. Another way is mechanical sharing, which is by we give mechanical forces to the genomic DNA, and this will cause fragmentation. Then we use molecules which are called adapters. These adapters they form sticky ends, and adapters are usually synthesized, so they form sticky ends with the restriction enzyme site of your interest. So they can then be ligated into a vector, right? This is what we can do. Then we go into transformation. As I already told you, transformation can also be performed. So in case of transformation, these clone is inserted into the vector. This process is called transformation, right? And it could be a bacterial strain. As I told you, there are several E. coli strains, um, other strains as well, or they are engineered for the specific function of your protein, or there could be yeast as well. There could be mammalian and insect cell lines as well. So talking about different forms of vectors, as I told you that vectors are essentially chosen. I'll just make this as a table based on the insert size. So I'm writing the insert size in megabase pairs. Um, So let's say plasmids are good for, let's say, 15 megabase pairs, lambda phage for 25 base pairs, megabase pairs, cosmids around 45 megabase pairs. So cosmids are basically a combination of plasmid and phages. Then we have bacteriophage, 120 to 300 megabase pairs. Then we have bacterial artificial chromosome and yeast artificial chromosomes, which express in bacteria or in yeast, which can take around 120 to 300. Sorry, this would be 70 to 100. I wrote it wrong. Yeast artificial chromosomes from 240 megabase pairs to around 2000 megabase pairs. So yeast artificial chromosomes are also used. But the larger the fragment size, the lower will be the efficiency of producing a positive clone. That's usually the problem. So again, if we look at construction of the schematics again, um, cDNA library. How is a cDNA library produced? Let's look at it. So let's say you have this is a bacteria from where you want to get the cDNA library from. It has its own genomic DNA. So what you want to collect is collect the mRNA, right? And now you have the mRNA in a tube, right? Um, this is usually performed using chloroform um, and isoamyl alcohol process. Then once you have the mRNA, you can go for reverse transcription. You will get the cDNA. cDNA duplexes. And then you will insert the cDNA into bacterial plasmid. So now these are plasmid with your inserts, let's say, right? And then you will insert this plasmid into bacteria. And as I already told you, this process is called transformation multiple times by now. So you will have bacteria with this plasmid inside. So there will be multiple bacteria which will have this plasmid inside. So now you will grow these particular bacteria. So they will grow in number based on, and they even the plasmids will grow. Obviously, they will have the plasmids. And how to only allow these bacteria which have these plasmid to grow? Usually these plasmid have a selectable marker, let's say an antibiotic resistance gene. So for example, it might have a ampicillin resistance. So the bacteria which will have your plasmid will be resistant towards ampicillin. So when you grow it in a media which contains ampicillin, only the bacteria which have the plasmid will grow. So that is how you screen it. Then you isolate the plasmid, and then you get the DNA basically. Now this DNA is essentially the DNA which will produce your enzyme. You can go for sequencing further to verify whether the right sequence has been inserted. Now, talking about the mRNA structure, how does it look like? So the mRNA structure usually has a 5' cap. Then there is a 5' untranslated region. This is not translated. They do not produce protein. There's a coding sequence in between. Okay, and then there is a 3' UTR. And then there is a poly A tail. Been as long as 20 A's. So this is the poly A tail. and methyl cap usually. So these poly-A tail and methyl caps are usually important for the mRNA stability. Otherwise, mRNAs, they have a very short half-life. They are usually degraded if they don't have these things. So how to isolate mRNA, how to do that process, that also we will discuss. So let's say you have different bacterial colonies, or you might have a tissue which you need to, first, if it's a tissue, you can homogenize it using a homogenizer. And then you use a lysis or a binding buffer, right, for this. And then you perform this binding in oligo-DT beads. So basically, based on the very characteristic that all these mRNAs have a poly-A tail, if you have a continuous T, then this A's will bind with the T, and this is the exact principle based on the the complementarity of genomes. We know that of DNA, it will actually help in binding. Then we will wash with buffers. Washing is a step, and then we will perform elution, and then we will collect something which is the poly-A tail plus mRNA, what we have. We will collect this in the after elution. And after elution, how we separate other, DNA and other kind of materials that are nucleic acids which are present in the in a cell, we actually perform centrifugation, right? That is the process overall. Now, talking about the step 2 of cDNA synthesis, how is it performed? So there are two ways to go about it. First, we can go for homopolymer tailing. So what happens is, because we already told you that RNAs have poly-A, so a T would be complementary to it. So this is how an RNA looks like, and there are multiple A's actually on the 3' end. And this is a mRNA. We want to create a cDNA. We will use a reverse transcriptase enzyme, and we will supply 4 DNTPs. Now, these DNTPs are basically DATPs, DTTPs, all the 4 essential uh, components of a DNA, the bases, nucleotide bases. So now we will have a mRNA with the cDNA, which will have corresponding Ts, and it will be synthesized using the reverse transcriptase, right? After this, homopolymer tailing comes into picture. So what we do is, in this DNA, is a cDNA strand. In this cDNA, we will add, let's say, cytosine to one end. And then we will perform alkaline sucrose gradient. It has two functions. One is to hydrolyze the mRNA. It will remove the mRNA, and it recovers full length cDNA. So now you only have the cDNA here, CCC, and then you have the cDNA on the 5' end, which will have T's obviously. And then you would add oligo DG, which will be complementary to this C's, and you will get actually the duplex form of the DNA. G then. Then this cDNA can be inserted into a vector, as we already know. Okay. Following this, there's another method for cDNA synthesis. It's called the Gubber-Hoffman method. How do we produce cDNA with this? So we have the cDNA, we have the mRNA, which has a cap and a poly A tail. We will add oligo DTs, and then this will essentially, help formation of. So there are A's, there would be T's, and if we add the reverse transcriptase enzyme and the DNTPs, we can actually go for the synthesis of cDNA. So now we have mRNA and the cDNA, one strand of cDNA, and we remove this mRNA using an enzyme called RNase H. We give some random primers because we don't know the sequence. So random primers are given and DNA polymerase. So what happens? Essentially, the mRNA is firstly removed and you have the DNA only with T's attached to it. The random primers will actually go and bind, and the DNA polymerase will cause polymerization. Now you will have something as the duplex of cDNA. This is a cDNA duplex, and then this can be then further inserted into a vector. Okay, so this is all about cDNA synthesis, how to screen. So screening is performed by DNA sequencing. Like the Sanger sequencing can be performed to see whether your clone has the sequence of the gene, right? There could be expression of protein, which will essentially involve some immunogenic steps. Let's say, for example, Western blotting, where you have antibody against your protein, which will help you to determine if your protein is getting expressed or not. Third could be the enzymatic activity. So for example, if a protein is having a function of a hydrolase, for example, lipase activity, you would actually check whether the protein that you are getting also has that activity. So that's also one kind of method. Screening can be done using DNA hybridization. What is this method? Let's look at it. So in case of DNA hybridization, it relies on the complementarity of DNA. So A binds with T, G binds with C, then there's a double and triple bond involved, right? We all know this. And based on this, we synthesize probes, and we will talk about how probes are synthesized. So what essentially we do in DNA hybridization, there is a master plate which has different colonies, right? This is called the master plate. And all these dots are basically bacterial colonies. And then you transfer the cells into a nitrocellulose membrane. And, you know, you create a replica plate of it. Replica plating is this is called. So now you'll have a replica plate. Then you will go for cell lysis, denature the DNA, and bind to the matrix. So matrix is basically this other region apart from the colony that is called matrix. So there would be bound DNA to the matrix very close to the colonies only. So you see the DNA is now outside the cell, right? And then you synthesize the probes. These are probes which have a complementary sequence to your DNA, and it will go for hybridization basically, where this complementary sequence of DNA will bind with your sequence. So let's say this is the positive clone. Here the probe will bind. And you know that this is the positive clone. So when you know this is the positive clone, you can take the master plate, you know this is the positive clone, you will go and isolate the DNA from here, and you will go for the further steps, right? These probes are usually radioactive. There could be also fluorescent probes and other forms of probes also, which can be used, right? Now, how are radioactive probes synthesized? So radioactive probes can be synthesized using a random primer method or uh, terminal transferase method. We'll talk about both of them. In case of random primer method, what happens? You have a DNA, let's say, and then you go for denaturation. What is meant by denaturation? It will basically take these two strands apart. It will break the hydrogen bond in between. And here you will add the oligonucleotide primers. These are random primers and they will perform hybridization, right? This is the concept. And you will, so if this is 5 prime to 3 prime and this is 5 prime to 3 prime like this, you will have several primers binding at separate locations. Similarly, in the other strand also. Then you will go for DNA synthesis with Kleno fragments. And you will add the 4 DNTPs which are required for DNA synthesis. And here you will also add the radio labeled probe. So here what you will get is all these probes which are attached to the DNA will have a radio label probe attached to it on, let's say, the 5 prime end, right? And probably then you will go for denaturation. And you will isolate these probes. So now you have synthesized these probes. These are labeled probe DNA. The other method, as I told you, is the terminal transferase method. What happens is, again, you have a DNA, 5 prime N, 3 prime N, 5 prime N, 3 prime N, joined by the hydrogen bonds. You give a lambda exonuclease. So that will essentially cause sticky ends. So from the 5 prime end, so you will see the 5 prime ends are resected, right? And then you will add a terminal transferase and radioactive ATPs. They will add A's, obviously, and they will be labeled, radio labeled. Because obviously you're adding radioactive A's. So how to perform screening? We already talked about it. So for screening, you need to perform, make replica plate. I think we already drawn the diagram. Then we go for blotting into a nitrocellulose membrane. Then we go for hybridization, where these probes will bind to your DNA of interest. And then we develop the blot. So if, let's say, we are using autoradiography, then we will perform, if we are using radio labeled blots, then we will perform autoradiography. Autoradiogram is an instrument which will actually tell you. There could be immunological methods for screening. How is it done? So again, the same concept where you have the plate, this is the master plate, you create a replica plate. Then you go for lysis, and the protein binds to the matrix. So your plate will now look something like this. Wait, it's difficult. So it has all the proteins here. I'm just drawing it in a very let's say your proteins, whatever proteins are expressed of in each colony will be here. So what you do now, you add a primary antibody. So primary antibodies are like this Y-shaped, and they are specific towards your protein, right? So this primary antibody will bind to your protein. Let's say here your antibody binds, right? After this, you give a secondary antibody. This antibody usually has, again, a label to it, like it could be a horseradish peroxidase HRP label. So any kind of label so that you can easily detect it. And it binds to your primary antibody. So after each step, obviously washing is very important, because if you do not wash it, then there's a problem. So the secondary antibody will bind to all the antibodies which are present. So you need to wash. For any immunogenic technique, washing is very important. So in this, if here the primary antibody was there, here the secondary antibody will come into picture. Again, you will go for washing, and then you will get the positive colony. So you know which is the positive colony. If this is the positive colony, then you go for, direct further steps with the positive colony from the master plate. Screening can be also done using enzymatic methods, and these are especially important for enzymes or proteins that have some enzymatic function. Let's say we are isolating hexokinase, which has an function of converting glucose into glucose 6-phosphate. So again, in this step, we have the master plate. We create a replica plate. We transfer this into a nitrocellulose membrane. We go for lyse the cells. So that will extract the protein. And you will also add substrate. Let's say you add glucose in this case. And then you will screen the colonies. So let's say this colony turns out to be positive, which can actually convert. So you will take the same colony from the master plate and go for further process, further purification or further overexpression process. So once the position of clone is known, then it is extracted from the master plate, as I already told you. The clone, which is essentially a DNA, is diluted for homogeneity, because it will not only have your own DNA, but the DNA of the bacterial DNA and everything else. So you need to dilute it. And then we need to check the purity using a PCR. So let's say this is the DNA, it is present. You perform PCR to check whether you are getting multiple copies of this DNA. That is very important by using sequence specific primers. Now coming to the part of post-genomic era, if a gene sequence is known, then the process becomes very simple. We simply, if a gene sequence is known, we take the gene, perform PCR, that is polymerase chain reaction, and we'll discuss about it in details. We will get the fragment of the gene, and then we will proceed to cloning as we did earlier. So what is a polymerase chain reaction? It basically amplifies double-stranded DNA and by enzymes. And we'll talk about what kind of enzymes are important. If you're not sure what a DNA is a nucleic acid which has uh, two complementary building block uh, chains, two complementary chains. So if you look at the structure of a DNA, this is how it looks like. It's a double helical structure. One strand is 5' to 3', and the other strand is 3' to 5', right? This is how a DNA looks like. They are formed of nucleotides, which are the smallest element in a DNA. They form from nucleotides. Nucleotides contain a phosphate backbone or phosphate group. They have a 5-carbon sugar. In case of DNA, it is a deoxyribose sugar and a nitrogen base, right? These nitrogen bases can be either purines or pyrimidines, classified into as the number of rings they have. So purines have 2 rings, and the purines are adenine and guanine. These are purines. Pyrimidines only have 1 ring, so they are the other two, that is cytosine or thymine. And we already know the base complementarity rule of DNA is that A bonds with T and G bonds with C. Uh, and these bonds are actually hydrogen bonds, right? Um, So we know that DNA is complementary in nature. So there is a primary strand, there is a which acts as a template strand, and there is another strand which is complementary to it, which is called as the coding strand. So these all things are just some basic concepts. For in case of replication, based on which PCR is developed, there are certain enzymes which are important, like helicases. These helicases, they unwind DNA and they will essentially unwind DNA and separate the DNA into two separate strands, right? There are primases. Primases forms primers. They form RNA primers in your cell. But in our application, when we do this in a cell-free method, in a PCR tube, we usually use DNA primers. But in your cell, RNA primers are produced. Primases are the enzymes responsible for it. There are single-stranded proteins, binding proteins. These single-stranded binding proteins prevent the single-stranded DNA to form double-stranded DNA. So let's say in case of a replication fork, this is how it looks like. So there will be several single-stranded binding proteins which will stabilize this single-stranded DNA structure. It is very important. Otherwise, we cannot go for replication if it again, if it opens up and it binds again, it is not possible. Fine. Then comes the most important player, the DNA polymerase, which will perform the polymerization. And then we have some tethering proteins, which stabilizes the polymerase. Otherwise, the polymerase can, fall off from the site. It is possible. To prevent that, there are these tethering proteins. So for DNA synthesis, the main steps are, initiation. So initiation, for that, you require a primer, right? And then there is the elongation step, which is also known as the synthesis step. And then the termination, that that it needs to be stopped. Initiation is also important, like the origin of replication, usually OREC sites in E. coli. It's there where it tells you this is the place from where DNA replication will start. In eukaryotic system, there are multiple origins because such a large DNA cannot be synthesized from a single origin. It will take a lot of time. So if we look at some history for PCR development, we'll come across certain timelines. In 1950s, DNA replication in cell was actually found out by Arthur Kornberg. In 1976, the thermostable DNA polymerase from Thermus aquaticus, known as Taq polymerase, you must have heard about it. If you have a very little bit knowledge of molecular biology, you must have heard about it. In 1983, Mullis, he synthesized DNA oligoprobes for sickle cell anemia, right? In 1983, again, repeated thermal cycling, which is basically the concept of PCR, was firstly used to clone a gene. In 1984, Mullis and Tom White, they tried amplifying a gene from a genomic DNA, but they could not visualize this amplification product in a gel with ethidium bromide. In 1985, the patent was filed for PCR and its application in specifically in sickle cell anemia. In 1985, again, Taq polymerase and BST, only these two were known, which were thermostable, and Taq was selected to be better, more suitable. In 1985 to 1987, okay, sorry, in the 1985 itself, in Salt Lake City in the US, there was the first demonstration or announcement of PCR technique. In 1985 to 1987, all the PCR components, which includes the instrument and the reagent, were developed, right? So what's a PCR again? It's a repeated cyclic reaction that mimics DNA replication. And we will see all the components which are essential for, PCR. The main steps are denaturation, annealing, and extension or elongation. Denaturation, basically, it will create double-stranded DNA into single-stranded forms. That is the step. Annealing will help in this single-stranded DNA. It will help the primers to sit in it. Okay, to base pair with the single-stranded DNA, the primer to base pair with it. This is the step of annealing. And in elongation, what happens is this primer is sitting and the polymerase comes into picture because polymerase cannot synthesize a double-stranded DNA from a single-stranded form. We require a primer for it. After that, elongation is performed. Okay, and in a PCR, let's say if you have 2 copies of a gene, let's say you start with 2 copies. After one PCR cycle, there would be 4 copies. So 1, 2, and it will produce new copies. After 2 PCR cycles, there would be from 4, it will become 8. No, sorry, it will become 16. So basically, after each step, the number of PCR products formed is 2 to the power n. But this is considering that there is ideal efficiency, which is not the case in case of PCR. So to find out the amount of DNA, the formula is C is equal to C naught 1 plus E to the power n. So E is basically the efficiency factor. This is the formula. C naught is the initial DNA concentration. C is the final DNA concentration. And N is the number of cycles. Now, if we look at thermal cycler, the functions, what happens in it, there is an initial denaturation phase, let's say 95 degrees for 5 minutes. Then there's a cyclic process. So there's 95 degrees for 30 seconds. Then we have annealing at 55 degrees for, let's say, 30 seconds. And then we have extension, which is based on the amplicon length. It's done at usually 72 degrees for Taq polymerases. This is for, let's say, 45 seconds. And this step is repeated 20 to 30 times, even more in some cases. And then you have the final extension step, let's say for 10 minutes or 5 minutes. And then you go for an infinite hold at 4 degrees. So what are the requirements of a PCR? One is the DNA sequence must be known. Otherwise, it's impossible to form primers. Then we, of course, require primers. These are short DNA stretches. The typical size is 20 to 30 base pairs. They are readily produced by commercial companies or can be performed, can be produced by DNA synthesizers as well. Then we have a thermostable DNA polymerase. Right? So, of course, the most important component is a thermal cycler, which will form this entire process. When we take a template DNA, which is again very important, template DNA could be from various sources. It could be genomic, it could be viral, it could be a DNA fragment. It could be from a plasmid as well. It could be a tissue sample as well. Now, there are various types of PCR that is beyond the scope of this course, but you can read about it as well. The basic idea is same behind all of that. In case of primers, There's a forward primer and a reverse primer which binds uh, and to the DNA, one to the template strand, one to the coding strand, and they are act as the starting point for DNA synthesis. Then we go for designing the primers. How do we design primers? The ideal primer length should be between 18 to 24 base pairs. If it is too long, then there is problem with the annealing step, because annealing is based on the melting temperatures of the primer. If it is too short, then we get non-specific amplification. The melting temperature of a primer is based on its GC content. Usually it's between 52 to 58 degrees, which gives us the best results. It could be higher, it could be lower. Sometimes, the DNA that you are getting all has a very high GC content. Usually this happens in thermophiles, in bacterias or archaebacterias, which grow in very hot environment. They have their DNAs with high GC content. In that case, you have to synthesize primers with high GC content. In that kind of situation, there are separate specific polymerases and polymerase buffers which help in mitigating this situation. How to calculate TM? Usually the most preferred formula is you simply add 4 into the GC content and 2 into the AT content, and you calculate the value in degrees Celsius. And this is actually good for primers less than 14 base pairs, right? But when it is the primer length is too long, then the formula changes. It's something like this: 69.4 plus 41 into G plus C, GC content minus 16.4 by the N. N stands for the length of primer. Again, then we have the annealing temperature. Usually it's 5 degrees lower than the melting temperatures. And the annealing temperature between the forward primer and reverse primer should not be greater than 5 degrees Celsius. Otherwise, there would be very poor annealing, right? The GC content should not exceed 60% ideally. And there should not be. So when you're synthesizing primers, so it's from 5 prime to 3 prime end. On the 3 prime, there should not be more than 3 C's or G's, because that causes a problem which is known as a GC clamp. What happens? This will basically bind to DNA with high GC content, and it will start synthesis. You will get non-specific bands. Although this can be solved with a higher annealing temperature, primer should not form secondary structures. So what are the different kind of secondary structures? There could be hairpins, there could be dimers, which are formed. There could be repeats. Okay, if repeats, they can cause problems. And there could be primer template homology other than the target site. The enzyme used for this is as we already know, it's stack polymerase, isolated from Thermus aquaticus. That's the organism. And it was first found in Yellowstone National Forest. It is also in US. It's a hot spring. Taq polymerase requires magnesium as a cofactor, very important, and its optimum temperature is 72 degrees Celsius. The disadvantage of Taq polymerase is it lacks proofreading ability. Proofreading is performed by 3' to 5' exonuclease. So proofreading is not present, so it has a lot of errors. So 1 in 10 to the power 4 error rate is there. So if you have a 400 base pair cycle, 400 base pair gene that you are amplifying, then after 20 cycles, the error would be 33%. So it compounds with the more number of cycles. And there would be random errors. For this, there are alternatives. So the alternative is PFU, DNA polymerase. It is isolated from Pyrococcus furiosus. That's the organism, and it has this 3' to 5' exonuclease activity, which is important for proofreading. The error rate is 10 folds lower than Taq polymerase. So we need to perform optimization in a PCR, right? So usually this is done using a gradient PCR, where the annealing temperature is kept as a gradient. So let's say this is the initial denaturation, then we get the denaturation, then we have, let's say, gradient from 52 degrees. So let's say 62 degrees. This is and there are multiple steps, multiple wells which will have, or rows which will have different temperatures. Then we will go for the extension, and then the final extension, and then the infinite hold. Some troubleshootings steps. So let's say there is no product formed, or non-specific, no product is formed, then you can check the template DNA quality, sorry. You can reduce the annealing temperature or simply perform the gradient PCR. Increase magnesium concentration, magnesium ion concentration. You can add DMSO up to 10%. So what does DMSO do? It prevents formation of secondary structure. You can use other thermostable polymerases, which are more efficient. And sometimes even, you really cannot explain what is the problem. It might be a problem with your primers, happened with me as well. So you throw out the primers and prepare fresh stock. And sometimes you don't know why this happens, but this happens. It's a common problem. Sometimes what happens, you get extra bands. So in that case, the steps are usually exact opposite. So this is essentially non-specific amplification. Here what you do, you increase the annealing temperature. reduce the magnesium ion concentration, reduce the number of cycles, or you can again go for different enzyme. For analysis, you load this this amplified product to a agarose gel. You go for ETBR staining, and then you visualize in a UV trans illuminator. The advantages of PCR are its speed, its ease of use, sensitivity, and robustness. Usually for PCR, the lower amount of DNA is more preferable. Less is more, usually in case of PCR. The limitation is you need to know the DNA sequence of the target for for primer designing. There is infidelity in replication process. So like Taq polymerase, it does not have the proofreading ability. Up to 5 kb, everything looks fine. But if you have more than 100 kbs, then it becomes a problem. Very large fragments cannot be amplified. It cannot be used in genome sequencing for multiple reasons, actually. So there is misincorporations of errors which can come in. So you don't know whether that is a mutation or if it is there due to PCR. So if there is any error, that will be amplified as well. Those kind of things become a problem. So because genome sequencing is done for checking, if there are mutations. But if these mutations are due to PCR artifacts, you cannot really verify that. So what else do we have? I'll I'll draw the schematics for cloning. So here you have the genome, let's say, from where you perform a PCR and you get your gene. You cut this gene with restrictions enzymes to get sticky ends. So this will be RE1, this will be RE2. Similarly, you will also have a plasmid. You will again digest this plasmid using restriction enzymes, and you will get something like this. You go for alkaline phosphatase treatment. This will prevent this plasmid to recircularize itself. Then you will basically perform ligation, and it will create a recombinant plasmid. Then you will perform something called as the transformation. So this is the bacterial cell with genomic DNA. It will also have your plasmid of interest. And then you get several colonies. You will perform screening to get your colonies of interest. So in cloning, what are the enzymes that are required? One is the restriction enzyme, as I talked about. There is the polymerase for gene synthesis. There is the alkaline phosphatase, and then the ligase. I'm talking about restriction enzymes. They come from the RM system, the restriction methylase system, which is a defense mechanism in bacteria. Usually, foreign DNA is unmethylated and they are cleaved by restriction enzymes. Self DNA of the bacteria itself are usually methylated and they cannot be cleaved. So that's the exact idea behind this. If you look at the nomenclature of restriction enzyme, let's say EcoR1, so it's named as E stands for the genus, O stands for the species, R is the strain, RY13. So from there we get the R, and 1 is the first restriction enzyme. It was isolated from this bacteria. So RM system can be like type 1. So type 1, they have recognition site, which is 3 to 4 base, 3 to 4 nucleotide at the 3' end. Then there is a 6 to 8 non-specific nucleotide. And then there is 4 nucleotide at the 5' end. And the cleavage site is 1000 base pairs away. Then there is type 3, which is not usually ideally used for molecular cloning. There is type 2, which is used. The cofactors in this case is S-adenosyl methionine, ATP, and magnesium ions. In type 2 restriction site, they are usually 4 to 8 nucleotides long. And specifically, we use type 2P. So they have palindromic sequence. And they cleave at the site of restriction, site of recognition, sorry. That's the idea. So for example, in case of BAMH1, the restriction site is GGATCC. So if it reads like this, in the opposite strand also, from 5' to 3', it will read the same, GGATCC. And that is why it's called palindromic. It produces sticky ends usually, although blunt end restriction enzymes are also possible. After this, we will talk about vectors. So vectors could be cloning vectors or they could be expression vectors. The idea is cloning vectors will amplify the DNA. Expression vectors will actually express your protein. So a vector should have the ability to carry foreign DNA. And it should have the ability to replicate in host. So vectors can also be designed based on, where it can actually express. So if you're talking about expression systems, so there are promoters in a vector. So anyway, we'll talk about it. So it should have a low molecular weight because low molecular weight can be easily transformed. It can get inside the cell easily. It should give some phenotypic changes upon transformation. So for this, we have the selectable markers, the antibiotic resistance genes, and all this kind of concepts. It should have a multiple cloning site where you will clone your insert, and it should also have a high copy number. So if we talk about the components, there should be an origin of replication. So let's say if you want to replicate it in a E. coli cell, it should have ORI-C. It should have a selectable marker, which could be like ampicillin resistant gene or kanamycin resistant gene, something like that. It should have a promoter. This promoter expresses all the genes which are essential, right? And then there are host-specific vectors, like say bacterial expression vectors are like PBR322, Puck series vectors, which are again modifications of PBR series. Then we have phage-based vectors. We have yeast vectors and mammalian vectors. So on the top of my head, mammalian vectors could be like PEGFP, pcDNA, something like that. These are all kind of vectors which can uh, be used for mammalian system. Then there are shuttle vectors. Again, these are a little bit out of the scope of the syllable. I mean, this course, shuttle vectors can replicate in multiple hosts. Okay, you can look after all the other different forms of vectors, but let's go with the questions. So the number of DNA duplexes obtained from one DNA duplex after 4 cycles of PCR is A8, B4, C32, or D16. They've already told you the formula is 2 to the power n. Here n is to the power 4, so it would be 16. Primers used for the process of PCR are single-stranded RNA oligonucleotides. They are single-stranded DNA oligonucleotide, double-stranded RNA oligonucleotide, or double-stranded DNA oligonucleotide. Usually in human cells, single-stranded RNA oligonucleotides form primers, but for PCR applications, we go for single-stranded DNA oligonucleotide. They are cheaper and more stable. The most effective technique for enzymatic amplification of specific gene segments of DNA is DNA hybridization, nucleotide sequencing, polymerase chain reaction, or reverse transcription. The answer would be PCR. The enzyme that creates sticky ends during homopolymer tailing is alkaline phosphatase, polynucleotide kinase, DNA polymerase, or terminal deoxynucleotide transferase. Okay, the answer to this is terminal deoxynucleotide transferase. Ligation takes place between adapter and linkers, linker and vector, 5' P terminus and 3' OH terminus, or the adapter and linker. As we already know, that ligation will happen if there is a 5' phosphate and a 3' OH. So this is the C is the right answer. The DNA ligase enzyme used in genetic engineering is a bacterial ligase, T4 ligase, yeast ligase, or Pseudomonas ligase. So mostly T4 ligase is used more because of its cofactor requirement. ATP is the cofactor which it requires, also with magnesium ions. Um, so based on that, T4 DNA ligase is used. For bacterial ligase, we require NAD+. We have already discussed about it in my first lecture itself. You can go back to those slides and look at it. DNA ligase mediated reaction during DNA replication causes addition of new nucleotide at the leading strand, addition of new nucleotide at the lagging strand, base pairing or formation of phosphodiester, base pairing of template and newly formed DNA strand, or formation of phosphodiester bond between 3' OH of one Okazaki fragment, 5' OH on the lagging strand. So in a DNA replication, for joining Okazaki fragments, that happens when there is 3' to 5', because there is polymerization activity is only from 5' to 3'. Okay. So whenever there is 3' to 5' DNA, there the synthesis is pretty normal. It's continuous. But when there is 5' to 3' DNA, then fragments are produced. These fragments are called Okazaki fragments. And they are joined using DNA ligase. The RNA strand in the RNA-DNA hybrid is removed by RNase H, nuclease, or phosphatase. We already talked about this in the Gerber-Hoffman method. RNase H is used. The dash endonuclease cleaves both single and double-stranded DNA molecule in a non-specific manner. It is a DNase 1. The following types of enzymes are responsible for removing individual nucleotides from the end of DNA molecule. Is it ligase, exonuclease, endonuclease, or modified enzyme? So exonuclease is the answer to this because uh, of the name itself. So exonucleases cleave from the end, whereas endonucleases cleave from between. The type of bonding present between N1 of pyrimidine, this would be, and deoxyribose of DNA is N-glycosidic bond, covalent bond, hydrogen bond, or phosphodiester bond. It would be N-glycosidic bond. The difference between sugar molecules of DNA and RNA is the absence of nitrogen as at 3' at 3C, presence of nitrogen at 2C, absence of oxygen at 2C, or presence of oxygen at 3C. So DNA stands for deoxyribose ribonucleic acid. RNA stands for ribonucleic acid. Difference is only in the sugar backbone, in the sugar, and it is based on the absence of oxygen at the second carbon. The polymerase utilized for PCR is extracted from Homo sapiens, Thermus aquaticus, E. coli, or Saccharomyces cerevisiae. The answer would be Thermus aquaticus because they are thermostable. All other polymerases do not function at such high temperatures. Antibiotic resistant gene present in PBR322 is tetracycline, kanamycin, gentamicin, or ampicillin. So there are actually two genes present here. One is tetracycline, another one is ampicillin. The enzyme which separates two strands of DNA during replication is gyrase, topoisomerase, helicase, or DNA polymerase. So gyrase is also a class of enzyme which comes under DNA topoisomerases. Um, the function of gyrase or topoisomerase is uncoiling of DNA. So when the DNA is present in a cell, it's in a supercoiled form. This uncoiling using multiple cuts is performed using gyrase or topoisomerase. Helicase is the enzyme which separates the two DNA strands, and DNA polymerase synthesizes DNA. So with this, we are in the end of today's interactive session. Thank you all for joining. I'll be sharing the slides as well as the recording with all of you. And if there are any doubts, any questions, you can definitely ask me. And there is also a feedback form that has been released to, it's a feedback from your end, whether these interactive sessions are actually useful or if there are anything that you need help with, you can actually give your feedback over there. With that, I think if there are no further questions, we can end today's session and we will meet again next Monday on same time, 6 to 8 p.m. Till then, I wish you all good luck and have a nice time enjoying the course.